Hello, everyone. Thank you very much for attending to this last seminar of 2021. And today we're going to have to Raúl de la Mata Pombo. Dr. Raúl de la Mata Pombo, after he has studied at the University of Vigo, he defended his PhD thesis at the Biological Mission of Galicia, one of the organs of the Spanish Research Council. And later on, he has been moving between working in forest genetic improvement programs and also addressing basic science questions in experimental infrastructures that these programs based in the USA, Canada, Sweden, and Spain have been providing to him. Right now, he is hired as a postdoctoral researcher by Xavi Picó here at the EBD in the Biology of Plants group, and he is now working with Arabidopsis plant evolution. So let me thank you again for coming to speak to us, Raúl, and I'll leave you the word.
Thank you so much. So, first of all, thank you to the organizers for giving me the opportunity to show part of my work, and thank you everybody for being here and people that's online. Today, I'm gonna talk about microevolution in forest trees. So, as many of you know, microevolution is this real-time evolution at a shorter time evolution. Actually means the evolution at population level, right? How populations adapt to their local environment and how populations diverge if they are living in different environments, right? This is the kind of evolution we are going to talk today. And the kind of organisms that are the goal of the studies I'm going to show are forest trees. Also, within the field of microevolutionary patterns, I selected 3 different topics because their relevance and because they are still open to intense scientific debate nowadays. And they are the selection patterns and the genetic variability, how it's distributed in natural populations, trade-offs, mainly life history trade-offs, and phenotypic plasticity. So the outline, the first point, I'm going to do the rationale about the core concepts in microevolution, these relevant evolutionary topics. Thereafter, I'm going to show a little bit of forest trees because they are unique organisms and it's relevant in order to address these kind of questions. And finally, I'm going to show results of 2 study cases. So first of all, let's talk about genetic variability and selection patterns, life history trade-offs, and phenotypic plasticity. Whatever natural population has variability in phenotypic traits, let's say body weight in a bird, right? The population, we are going to find individuals with heavy individuals, lighter individuals, and most of the population is around the mean, right? So not all the phenotypes in a population are the same. And actually this phenotypic variation may have a genetic control, right? So this variation can become genetic variation. So what theory predicts? Actually, if the mean of the population is not in the fitness optimum, natural selection will push the mean in the direction of the highest fitness, right? This is just what the theory states. But as consequence of this natural selection, when it's directional, when there is not balance in selection because, again, the population is not in the optimum, the consequence is a depletion of genetic variability as a result of the selection. Actually, this is known as the Fisher fundamental theory of natural selection that says selection depletes additive genetic variation and change the population mean in fitness-related traits. I mean, this is very easy to understand, this concept, but actually Darwin, in the chapter 4 of The Origin of Species, talking about natural selection, states that this is a slow process, right? This is maybe because all of us, we have this idea of the selection pushing populations progressively, right? But is this right? Actually, after the last 50 years, science has been gathering results from evolution from a quantitative perspective with mainly bird populations, for example, mammal populations. And in the last decade, there's a lot of work, meta-analysis, and reviews using this data set. And what is the data set showing? First of all, has been detected widespread directional selection. Looks like the natural populations are not in the optimum, are not in equilibrium with the optimum fitness. They are still moving in one direction or other. And this is widespread in nature for many kinds of organisms, right, and many kinds of systems. But what has been also found, that there's what is called phenotypic stasis, has not been detected a big change in trait means in the populations. There's a lot of genetic variability out there in spite of strong selection pressures. What has been found also? Yes, this is, here you can see many relevant scientific publications showing same results, the large amount of genetic variability out there. So we have a problem here, because the theory predicts that is natural selection, and actually it has been found, directional natural selection, we should be expecting depletion of genetic variability, and we should be expecting a movement of the trait means. But it has not been found in this kind of long time studies, right? So, and of course there's hypothesis of why it is happening, right? Of course, mutation selection balance, a lot of mutation in the populations, creating new genetic variability. Fluctuating selection in space, I mean, populations are not homogeneous, so individuals in the same populations, they can be under different selection pressures. Fluctuating selection across time. Today we are going to see some results about this question in forestries. Also, migration. Life history trade-offs, they create variability and can hinder the movement of the trait means, and other kind of genetic constraints, right? They are hypotheses that can be explaining these results. So fluctuating selection over time is an easy thing to understand why there's not a depletion of genetic variability, neither there's a movement of the trait means. One of the first works on this question are Grant and Grant study about Darwin finches, when they detected after 30 years actually of this study, is that the beak shape of the birds is selected for based on different characteristics. Some years long and narrow beaks were selected positively, but other years wider and shorter beaks. And why is this? It's because the food source that depends on the kind of plant that is dominating the ecosystem in these islands, right? Different species of plants, different kind of fruits, and so the birds need different kind of beaks, right? And this dominance of one species or other in the island depends on precipitation, right? So finally, precipitation is controlling somehow the beak shape of these birds. But importantly, because some years, some phenotypes are selected for and are different from other years, all these shapes are kept in the population, right? So here we have a mechanism to keeping genetic variability and avoiding the population moving in one direction or other. So is this common in nature? Probably yes, right? Actually, Ciepielski, in Ecology Letters, he has a couple of papers in the last decade actually checking if this is common It's a meta-analysis. And yes, it is. Selection changed a lot. So this idea, the Darwinian idea of a slow thing pushing, looks like it's not real. Actually makes sense because which exert the pressure is the environment, and the environment changed. So it's easy to understand that the selection changed as well, in direction or in magnitude, right, or both. So now we are going to talk about the trade-offs. There's this second topic that is under debate. Here we have what is called the Darwinian demon, right? Darwinian demon is a hypothetical organism that can maximize all aspects of fitness simultaneously. It's an organism that will reproduce directly after being born, produce infinitely many offspring, and live forever. I mean, it's just an example that this kind of organism cannot exist. And here we go to the core of the trade-off ideas, and the trade-off hypothesis is a core part of the life history theory, right? This shows that here is an example for a plant, but maybe the same for animals. Resources, here water, light, nutrients, are limited in nature, usually limited in nature. So the organism has to allocate these resources to different life history functions. Let's say reproduction, growth, and resilience, defenses, drought resistance in plant, whatever. So because the resources are limited and the organism cannot allocate all the resources at the same time, has to choose somehow. And this choice can be under genetic control, right? So actually the trade-off is defined as a negative association between two or more traits, being the traits under positive directional selection. I mean, it could be great to maximize all of that, but it's impossible. So finally, it's a strategy for an organism where to put the resources. For example, let's think in a tree. For a tree to grow, thereafter we are going to talk a little bit more, is an investment for future resources. But the tree has to defend itself from pests, diseases, herbivory, right? What's better? If the tree invests in defenses and is not attacked, makes no sense, right? So it depends of the context, the environmental context, right? Finally, this strategy of allocating resources becomes a strategy, and the strategy is under selection. So this maintains variation because there's not just one strategy to maximize fitness. What's better? Depends, firstly depends, and probably the best strategy is an intermediate allocation. Whatever. These trade-offs is other mechanism to keeping genetic variability within populations. And actually, because the trade-offs are context dependent, because if a tree is attacked or not depends many times of random circumstances, so this gives even more genetic variability within populations, right? So today we are going to talk a little bit about trade-offs as well. What's the open debate? In the literature, detecting trade-offs and life history trade-offs is not easy. It's not easy because correlation doesn't mean causation, right, firstly. And there's, for example, a general vigor problem. To detect trade-offs, we have to compare organisms with the same resources, right? If one organism invests more in growth, is more vigorous, maybe if the strategy works, it's going to get more resources. So we cannot compare individuals with different level of resources, right? We need to compare individuals with the same level. Actually, genetic correlations is the best way to demonstrate trade-offs using sibling analysis, and the best thing to detect trade-offs is correlate the phenotypic traits with the functional traits and with the resources. We have to be sure that two traits, they share the same resources. And finally, this other question, this other topic, phenotypic plasticity, in the last decade has been more and more relevant in evolutionary biology, right? What's the phenotypic plasticity? I mean, it's the different expression of the genes in different environments. Same genes, different environments, they express differently. So they produce different phenotypes, right? This is phenotypic plasticity. Still in literature, it's amazing because many times are confused, right? The concept of adaptation and acclimation. Acclimation is the plasticity. Same genes producing different phenotypes. It happens at individual level. It's fast, it's faster, and can go forward and backward. But adaptation is a change in the genetic makeup of the populations. What adapt are the populations, not the individuals, right? We have the genome, and this is almost the same, right? So adaptation and acclimation are different things, but they are part of the same game. Actually, plasticity has been seen for many years in breeding, but even in evolutionary biology, is like a noise, right? In breeding, it's a noise because you want to select individuals for a specific environment or for a region. And so in the test, you can see that in every single environment, you have a ranking of different individuals. I mean, there's not the best individual in all sites, right? This is a noise. But actually, plasticity is an amazing mechanism for organisms to increase the fitness, right? But from an evolutionary perspective, traditionally, it's also a noise because let's say that if I have genes that are not good ones, but I can express these genes that are not perfectly fit to the environment, but I can express these genes somehow that produce a phenotype that is fit, somehow is hindering the selection to lead this gene from the population, right? So this is the traditional perspective, but what's going on? Plasticity can be under genetic control. And the genetic control of plasticity actually is the famous G by G interaction, genotype by environment interaction. So if plasticity is under genetic control, that means some genotypes are more plastic than others, for example, or means that some genotypes have a plastic response in different direction of others. So if it has genetic control, can be under selection. So now there's a new perspective that plasticity is a trait by itself, that can be selected, can be promoted. Okay, I'm going to skip this. These are the reaction norms representing genotypes in an environmental gradient or in two sites, for example, for a phenotypic trait. These two cases are the cases of plasticity. Here is a crossover interaction effect. I mean, the plastic response change in direction, and here is just that change in slope. This is a typical, for example, in forest trees. In a low resource environment, all the genotypes are closer for growth, for example, but when the environment has good resources, there are some genotypes that are more sensible, they react more, right? So now the question is, okay, if phenotypic plasticity is a trait and is under selection, what environmental parameters triggers plasticity? I mean, select positively for plasticity, and which ones select negatively for plasticity? The theory predicts that there are some parameters in the environment that select for or against plasticity. First is environmental heterogeneity, right? Because plasticity is a trait that is helpful when the environment is heterogeneous, both in space or in time, right? Also the predictability of the environment, because if the organisms cannot predict what's going on, the plastic response can be maladaptive, right, or cannot be enough. So predictability is other of the parameters of the environment that can trigger or stop the plastic evolution, right? And resource availability. It's supposed that under good resources, the organisms can invest in a plastic response. And if it's not working, going back, right, in the plastic response. But in low resource availability environments, this investment is costly, right? Investing plasticity, if not working, is not a good deal. So we have, and actually all these questions has not been still deeply addressed, right? And this is relevant. So I already talked about these basic questions that I'm going to address in the study cases. But before, let's talk a little bit about trees. It's going to be short because there's no time for that, right? But what's going on with this kind of organisms? Actually, all these mechanisms and all this evolutionary debate works for whatever organism, right? But I've been working with trees my entire career, but not now with Xavi, that we are working with an annual plant, Arabidopsis. And doesn't change the hypothesis, doesn't change the rationale, doesn't change the questions actually. But working with trees, what changed is the experimental approximation, right? What are the main characteristics of trees? They are massive, right? They are redwoods in California, they are massive. And they live almost forever, right? For a tree, live 300, 400 years, it's easy. But this kind of trees, this species, maybe 5,000 years, something like that. Actually, the big size is a consequence of the long living, because the trees never stop growing. They can stop growing in height, probably because hydraulic limitation, but the secondary growth in diameter never ends. They always grow. This is why we can know the age of a tree counting the rings, right? So, but what consequences have these characteristics for the evolutionary processes? First, long generation times. We don't expect very fast evolutionary change in trees, right? Because generations, we don't have 1 generation per year. Also, because they live so long, it's expected that selection pressures change over the life of the tree. I mean, if a tree will live for 500 years, it's very easy that the environmental conditions will change over time as well. Also, there's 2 questions that are relevant in terms of the interactions. Because they live so long, finally, these interactions can be understood as very random, but also because they are massive and they live for so long, they are always there, right? They don't have strategies to hide. They are not cryptic in any stage. They are just there. Trees attract enemies the whole time. Actually, plant-insect interactions are supposed to be main drivers of evolution in plants, but in trees it's especially relevant, and they have thousands of mechanisms for defense themselves, right? So what's going on with growth? Why I'm going to talk about growth? Because this is the trait I use. This is the trait I use because timing things, I delete all the part of breeding programs, but in order to address these questions, I use the breeding programs, right? Because otherwise, to make this experimental work with trees is almost impossible, because it takes so long for them to grow, right? And what I use the tree breeding programs, but the data sets that we have in breeding programs are very reduced. The phenotype we have is just growth, survival, and this kind of things, right? Many times we don't have phenotypic measures about defenses or about this kind of thing. So growth is the trait I can use. But growth in a tree, in many organisms, but in a tree, is a trait that can be very close to fitness, right? Today we're going to see the results if it really is very close to fitness. Why is close to fitness? I mean, we know that faster growth increases the competitive ability. Trees, what they try is to get space, because if they get space, they get the nutrients, the water, and the light. And a forest mainly is a competition for the space. So to grow faster is an investment to get resources. Also, they go faster to the first reproduction, increasing fitness for sure, right? If there's a perturbation, a fire, a pest, to be able to reproduce, provide an advantage, right? And also, fast growth increase this early survival, right? When the tree is weak. When the tree is massive, they are very resilient organisms. When they are small, when they are seedlings, they can die. They can be just eaten or whatever, right? So being able to move from a seedling to a bigger tree faster can provide an advantage as well. But we also know that fast-growing trees or fast-growing families, genotypes, let's say, they have shorter lifespan. And actually, within the plant defenses theory, at least 3 of the hypotheses, the plant vigor hypothesis, the resource availability hypothesis, and the growth differentiation balance hypothesis of Matheson, the 3 of these hypotheses state potential trade-off between growing fast and defenses, right? So let's go with the first study case. This is part of my work with Dr. Anna Sala at the University of Montana, another postdoc at the lab, Sharon Hood. Anna is a ecophysiologist, Sharon is a fire ecologist. And this is a paper published in 2017. And what we did here, again, I took advantage of a ponderosa pine breeding program. Ponderosa pine is a pine of the Western North America, distributed all along the ranges, the Rocky Mountains, Sierra Nevada, the Cascades. These are 5-pronged ecosystems, right? So, and part of this system is this tiny insect. This is a bark beetle, the mountain pine beetle. You can see here, after an attack, how the tree tried to defend itself, producing the resin, like it's a physical barrier, but this resin is full of chemicals, poisonous for the insects, right? So here the question is, kill or be killed? If the tree is able to throw out the insect, maybe it can survive. The insect needs to kill the tree in order to reproduce within. It's going to try to reproduce in the phloem. This is the nutrient part of the tree and overwinter there. So this is a fight. How this is my finger, so this tiny insect, how it can kill the tree upon a massive attack. Actually, they coordinate with pheromones to say, okay, come on, attack the tree, overcome defenses, interrupt the system, so the tree is done. The first year we don't see it, the next year we realize that tree is done, right? Actually, thereafter, the insect, once there's enough insects in the tree to avoid interspecific competence, they produce a pheromone to say other insects don't come here because we are enough. And actually, this pheromone is being used to try to stop this, because this is a problem. In red color is this species, how it's going forward. Actually, this is Western Montana when I did the study. And Western Montana, as you can see, they are the Rocky Mountains, right? So the pest is now stopping the Rockies. Here in British Columbia, that the first economy is forests, timber, 5 million hectares has been killed by this insect. The thing, if it crosses the Rockies, it attacks ponderosa pine, but the preferred host is lodgepole pine. Lodgepole pine goes till here, and here is Pinus banksiana, is jack pine, and both pines, they hybridize. If this insect start attacking jack pine, can go all through the Canadian Shield to the east of Canada. So Canadians are really very concerned about what's going on. So what we know, that actually trees and ponderosa pine populations, they have genetic variation in defenses. Not all the organisms, not all the trees, they have the same capacity for the fence themselves, right? There's genetic control in the fences, and it's very variable. So this is Montana. This is the Missoula in western Montana. This is the Idaho Panhandle. This is Yellowstone National Park. This is Glacier National Park, beautiful. And I took advantage of a progeny trial of a breeding program. What's a progeny trial? This is a progeny trial. I mean, trees planted just there. But the important thing is they are from different families. Half sheeps, they share the mother, but not the father. In quantitative genetics, this is one of the pedigree schemes, right? So this is the trial, and as you can see, it was naturally attacked. For the breeding program, sometimes it's bad luck, but actually for these studies, it's perfect. We have a pest attacking our trial that has a experimental design with blocks and different families. So let's see if there's genetic variation here, and let's see other kind of things, right? So first question, does herbivory change the strength or duration of selection? There's fluctuating selection, right? Does this change arise because a growth-resistant trade-off? So what we have, in 74, trees were planted. So height, diameter, and survival, as usually in breeding programs, were measured during these years. At some point here, after 2001, the outbreak started. We don't know exactly the year because probably the outbreak started progressively, right? In 2013, I arrived to Montana, and we measured the trees killed by the insect. And using a retrospective scale, we were able to figure out what specific year of the outbreak every tree was killed by the insect, right? So we have survival and growth these years after plantation. This is the data set we have. So what we have, after 37 years or something like that, just 40% of the trees survive in terms of the ones we planted, right? But there's two different processes of mortality, before the insect outbreak and after the insect outbreak. And two processes, they have very similar rates of survival, right? That actually from a statistical point of view is very helpful. So there's a change in the selection. Okay, if we consider 2 times before the insect and after the insect, and we make a ranking of survival, actually we are representing the G by G. What we see is the families that are very resistant or less resistant are different, right? Can you see the change of the ranking? So this is the first result suggesting that there's a change here in the pattern of selection because we are representing survival. If we represent the genetic correlations of the accumulated survival from one period to the former, we can see that before the insect outbreak, correlation is almost 1. This means the kind of families that are dying are the same. But when the insect arrives, there's a reduction in the correlation. 0.7, given that it's accumulated survival, is a lot. Again, it's showing that the patterns of selection are being changed, and this is because the insect. So, change the patterns? Yes, they change. So, and in terms of selection, the direction, and the magnitude, for growth is a trait I can use. Here is a representation of the selection responses, means genetically, and selection differentials, means phenotypically, right? Differentials is in dashed line, responses in continuous line. So, I mean, here at the beginning, in many periods this is not significant because the percent of survival or mortality is reduced, the selection pressure is not big. But in a specific year, here we can see a positive selection for growth. This means that families that grow faster, they survive more. That makes sense, right? When they are seedlings, the families that grow more survive more. And actually, because growth is under genetic control, it's not just a selection differential, it's also a selection response. But then the beetle arrives, and the selection differential changes. Biggest phenotypes, and here are the trees, they have the same age. Biggest genotypes, phenotypes, they die more, right? And also it's a negative selection response, right? Here we have year to year. If we represent the outbreak as just a one selection process, we can see how the insect makes the selection differential so negative that brings a negative genetic response for growth. So if we see this from a cumulative perspective of selection, we can see positive selection for growth, negative selection for growth. So the genetic response is neutral, finally. We have an example of fluctuating selection that keeps genetic variability, but also keeps probably the trait mean the same. And this is why in all the breeding programs, the first thing the breeders do is to analyze the heritability of growth. And always, 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 there's a lot of heritability in growth. There's a lot of variation in growth that actually this may be explaining the thing, because to grow fast is not always good in fitness terms, right? So forests, they keep a lot of variability, and probably this variability has a role in the resilience of the forests. So yeah, there's evidence of fluctuating and opposing selection in these long-living organisms. I believe it's the first time to be shown and makes sense. So, but now this change has to do with the trade-offs. If we represent survival versus growth before the beetle as a period, a specific year, after the beetle over a period and a specific year, we can see here a negative relation. It's a genetic correlation. Every single point is a family. So survival and growth are negatively correlated, because survival there represent tree resistant to an insect. We have a trade-off. Fast growing families die more or resist less to the insect. So do we have here a life history trade-off or a resource-based trade-off? We don't know, because for that we should demonstrate that the families that die more, they have less defenses, and we don't have the defense data, right? But we know it's a trade-off, and probably life history, but not resource-based trade-off. That is actually probably the most interesting thing, right? So in this study case, main conclusions: there's a strong change of selective patterns before and after the beetle. Fast growth was selected during juvenile stage, but slow growth was selected under intense herbivory. If there's herbivory or not, if there's an outbreak or not, this is a random thing, right? We don't know, but these random things may have relevant roles in the evolutionary processes. Second, there's a growth versus tree resistant trade-off. So we have here two mechanisms to keep intrapopulation genetic variability: the opposing and fluctuating selection and the growth versus pest resistant trade-off. So finally, in 5 minutes, this is a paper we are writing right now, and this study case is about the phenotypic plasticity. Now, it's the same breeding program as before, but now we are going to use 3 different progeny trials, because we want to measure the plasticity, right? Same genotypes in 3 different environments. We have populations, it's not a wide range. It's just northern Idaho. This is the Panhandle, the old growth of ponderosa pine forest here, and western Montana. I mean, we are very conservative, because for these kind of purposes, it's better to use populations in the extreme of the ranges, right? It's easier to get results. But this is tricky, because we are comparing very different evolutionary trajectories. Here, we are comparing populations that are close with the same demographic pathway. But what we want to check here is environmental heterogeneity driving the phenotypic plasticity? And the resource availability is driving the phenotypic plasticity. Remember these questions we talked before about phenotypic plasticity. So this is a first result typically in quantitative genetics. These trials, they have population and progeny structure, means we have different populations and different genotypes within populations. So if we compare the population by environment variance and the family by environment variance, it's the first result I got. It's super weird. Why there's this thing and what means? Think a little bit. What means that? If all the G by G is accounted by the population by environment, means that populations, they have different plastic responses, but not the genotypes within populations. Means that genotypes within populations, they have a common plastic response, but they are different between populations. It's talking about local adaptation in plasticity. Genotypes from the same population, they have the similar plastic response. So it's a suggestion that here plasticity is being selected by selection, right? So the first thing I did is estimate the plasticity. Actually, I estimate the genotypic stability that is based in mixed models and reaction norms. This is the typical in breeding. In ecology, still, there's other approximations that is the difference of the means between environments. Still is very used, but in breeding, all the breeders, they use, and we use the slopes. And actually, at the first of the 20th century, was a big debate about how to estimate the plasticity. Because I'm a breeder, I'm on that. But this is other day, maybe we can discuss about this question. So I selected one of the models, and so I estimate the plasticity for every family, right? Some families are more plastic than others. So now I correlate with heterogeneity of the origin of these populations. And in order to do that, I use geostatistics. I'm not going to go in deep about that, but imagine these two different situations. Here is big patches, here is smaller patches. It's supposed that smaller patches will trigger more phenotypic plasticity, right? Because for an organism, the offspring of an organism, it's more difficult to predict in what environment it's going to live. So plasticity here may have an advantage, right? So what I use is just climate data around every population in a 20-kilometer radius, and I use geostatistics to estimate this environmental heterogeneity in terms of the patch size and the intensity of the variation with, among patches. And this is what we found. I mean, there's not a very consistent response, but we can see here is the plasticity, more plasticity, less plasticity, and here is patch size. So there's a general trend of more plastic population comes from environments with smaller patch sizes, what actually meets the theory. But the amplitude, the intensity of the difference of the environmental parameter within, among patches, there has not an effect at all. Actually, the only one that has a very relevant effect is the cold regime, where very plastic populations came from environments where cold regime is in tiny patches, right? So, I mean, we have here, these results are suggesting that yes, more heterogeneous environment makes more plastic population of these forestry species, even for populations that are very close, right? There's no big differences in the Rockies, but it's a mountainous ecosystem, right? And now, here I'm comparing for the same 8 climate parameters, the plasticity, again, a lot of plasticity, less plasticity, and here is the parameter mean, right? Is drought or heat. So the result is in warmer and drier locations are more plastic populations. It goes against one of the hypotheses because, as you remember, high resource environments should produce more plastic populations, but this is not happening here. Looks like stress is triggering plasticity, not against, right? And finally, just last thing, I mean, some results over the last year show that actually forests are suffering and are in decayment. So in terms of climate change, actually European Union, and this is a debate of, we are going to plant trees as a sink, carbon sink, right? So if actually this is going to be done, if humans we are going to plant trees, we have to should take into account all of these things, because the plants the trees we are planting now has to live for many years in the future, right? So resilience is a very important trait, not just productivity. And these results, what are showing is that somehow forests preserve a lot of genetic variability, and this genetic variability may play an important role in the resilience of these forests. And also, phenotypic plasticity can be an incredible mechanism for trees because they evolve slowly. But plasticity is a more faster mechanism. We have to take into account plasticity as a trait of selection. What I mean is, if we're going to plant trees, we should select the proper seeds, and one of the goals should be to make resilient future trees, right? So just what we found here. Ponderosa natural populations, they have potential to evolutionarily respond to defend from the insect. Also, ponderosa natural populations, they have potential to evolutionarily respond to abiotic stress through distinct acclimation responses. There's genetic variation in plasticity here, and we can take advantage of that. We can breed for resistance and increase plasticity. We can select families with increased plasticity if we want, right? And with increased resistance. And we have to take care, because artificial selection for increasing productivity may be reducing forest resilience through two mechanisms. First, we are depleting genetic variability, and it may have an important role in forests. And also reducing pest resistance, because if we select for growth, we are selecting maybe for reduced tree resistance to pests and diseases. And that's all. Thank you so much.
All right, thanks, Raul. Really interesting talk. I'm sure lots of questions for Raul.
Yes. Hi, thank you very much for your talk. Very interesting, very beautiful work what you have done. I was wondering, do you know about any comparative study that compares plasticity between organisms with different lifespans, like comparing short-lived plants, for example, with long-lived plants in terms of plasticity to, I don't know, for example, plasticity to drug stress or something like that, in order to see if there is a trend towards a positive selection of plasticity in plants with long living strategies.
Very good question.
Has this ever done, or?
Not in my knowledge. Checking for the bibliography for this paper that is written here. There's, oh, I don't have it. Alpert and Sims in 2000.
Sorry, again?
Alpert and Sims. They wrote a paper.
Okay.
But everything is on the. Oh, no, I have it.
Okay, you have it.
It's on the paper. Everything is on the paper. The hypotheses are there, and of course, lifespan may have a role, mainly because the speed of the response of the organism is if the speed of the response of the organism is too slow in terms of the speed of the environmental change, plasticity makes no sense. But the question is, there's comparisons, not in my knowledge. Actually, still there's not a good body of results in terms of plasticity for single species, so probably in order to compare species, there's not. Actually, maybe in a couple of decades we will gather enough results, as the same with the selection. Now we are producing the results of the long time series from the 70s, right? So in terms of plasticity, my point of view is because this new paradigm, still we don't have enough studies. I mean, if we take advantage of the breeding programs and the works in forestry journals, we can retrospectively somehow, all these G/G estimations, so typical in breeding, but we should change the perspective, right? So yes, from my perspective, until my knowledge, this studies doesn't exist, and actually there's not many studies relating plasticity with environment. There's in frogs in Sweden, there's a very classical work, and there's some others, but not from an experimental point of view. There's a lot about theoretical framework about this question. This is a very, very, very open field still. Thank you.
Yeah, well, I hope we're slowly filling that open field. So we have some frog studies here. We're also
Nice.
studying plasticity.
Nice.
So if there's any other questions, I'm gonna sneak in quickly, but then I'll pass on the mic because it's connected to this issue of plasticity. So I was interested in how in those final slides, how you actually measured plasticity. I mean, usually when we do it, it's with experimental setups. We have common garden experiments. And how did you measure that in the tree? Like, what was that axis? And then a related question to that, you talked a little bit about the slopes, the reaction norms, and I was interested for, you know, when you have organisms like trees that live for so long, surely that plasticity is not a single slope, a linear process, and you might have, for example, you know, once the trees combat the insect invasion, that then they might bounce back to faster growth, and whether that's somehow also included in your estimates of plasticity.
Thank you. Very good questions. One of your questions is methodological, right? Yeah. It's very, very used as index of plasticity. One index created by Fernando Valladares, right, is very used in ecology. That is a parameter that came from a difference of means in different environments, right? Here you have different models. They are based in linear mixed models, and it's about the regression line. The Sugla model, for example, is very similar to the equivalence concept in ecology. I mean, there's a lot of maths here and a lot of statistics. Not sure what's the specific question.
Well, I mean, because so how do you measure the variation in phenotype for a given genotype? I mean, without experimentally doing that.
These mixed models, they have a genetic part, environmental
Okay,
part,
so
and the G/G. The matrix in the linear model, you have a matrix with environments and the genotypic responses.
Okay, so it's genotype at the.
It's in a
population
matrix.
level.
So
Okay.
every single model has different assumptions. For example, Sugla makes a different value for every single family. This value of the Sukal parameter is a proxy of the plasticity. Actually, it's the opposite. It's genotypic stability, because breeders,
yeah,
they look for
about
the
it.
stability,
Yeah.
right? Because
Yeah.
if I want to plant this wheat or corn, they want the corn to work in all the environments. They look for the stability, right? And all these big matrices, and every single of the models, they have different assumptions, right? And the second question was?
Well, whether, you know, especially for long-lived organisms, I mean, it's unlikely going to be a linear,
It's because the trade.
unidirectional
It's because the trade.
response.
If you measure growth after 40 years, what you have is how to say, is integration of the growth of the tree every spring and summer. Every spring is you know, an accumulation. This growth was plastic, probably 1 year growth more, less, but measuring the final growth, you have a very good proxy of the growth potential of the family. So means the growth potential that is genetically controlled. So what I know is this family has big growth potential, right? Is what I want to know and correlate with the plasticity.
But so if it grows very fast early on and very fast later, I mean, then is that averaged, or I mean, do you lose that information if it goes up and down?
It's the accumulation.
Okay.
It's not the average. It's the accumulation on all of this growth, right? Other question is to see the within individual plasticity over time. A very good thing, but I didn't address this, right?
Okay.
Because maybe some families, they grow fast at the beginning, but thereafter stop. I mean, as a breeder, I know that. There's a juvenile-adult correlation that is very important. When to make the selection. For example, in Maritime Pine, the selection shouldn't be before 12 years old, because thereafter, because before 10 years old, these rankings can change. You are selecting the one you believe grows fast, and thereafter it slows. It can happen. But here I have an integration measure of the growth, what means that it's a proxy of the growth potential from a genetic perspective.
Okay, thanks.
Thank you so much.
So squeezing in a few more questions. I don't know whose hand came up first. Agus?
Hi, Raul. Thank you again for your talk. I'm concerned also with the loss of populations due to climate change, and so from the perspective of forests, what do you think would be a better technique to try and avoid this loss, this retreat for many species due to the climate warming? Do you think that we should start moving or using stands from the border, from the geographic limits of the species, to impregnate trees in other parts of their distribution, or would there be any other techniques that we could apply to do that?
Thank you so much. This is a very good question, and this is under debate in forestry management. That is called assisted migration, using populations from the south and move the population from the north. It's difficult to give an answer to that, but based on my results, I will do this rational. First, when we move a population to the north, what population we move? Because actually, we finally will move a sampling of the population. And we can see here there's a lot of variability within populations. So if we move things, but what proportion? Is not better to try or to wait that the population can respond naturally? Because we still don't know if they will be able to do it after a strong selection pressure. If we start to move things, and assisted migration in BC is the regular thing, moving things, because these transfer equations. So yes, maybe it's a good methodology, because this population now in the future will be fitted here, yes, but are you going to move enough genetic variability? A second, in these transfer functions, phenotypic plasticity never is taken into account. It's the same as in models of distribution models. Now, phenotypic plasticity is very incorporated to the models. This is why phenotypic plasticity is the future of the research in the next years, but they still are not in the models. So we know that even trees, they are able to plastically respond. And the models we are using to move things doesn't take this into account. Maybe we are moving a population that, yes, maybe is more adapted to the climate mean here, but it's a less plastic population also, and we are losing part of the capacity of the acclimation. And all of that is not in the model. So I believe there's too much things still open, but yes, assisted migration will be done, is being done, just we need more results and see if this is the only strategy we have to help forests to acclimate and adapt to the future. Thank you so much. Thank you.
All right, I think, yeah, we have time for one last question. Yeah, was there any? Sorry, I didn't ask. Yeah? All right.
Great. Thanks, Raul. Thanks for the talk. It's always nice to have people interested in phenotypic plasticity. I'm sure we'll have a lot to discuss. So you answered to Chris one of the questions I had, but so you have several trees that belong to the same family in different environments. That's your data set, right? And you have a good number of trees in each?
24,
okay.
But always over 18. For example, as a breeder, I know that in order to make a good estimation of heritability in half shifts in trees, 18 trees per family and site is a good number to reduce the standard error.
Okay, and the other thing, so how are they dispersed? Is it through like birds or their seeds?
In this species?
Yes. Because when you talk about the patches, the environmental patches, for plasticity to evolve is important the scale, whether it's fine coursed or like coarse grain or fine grained.
This is totally the point. Just for timing things, I didn't went through, but this is about how they distribute, right? How they spread.
How they disperse.
You are talking about the seed thing. Yes, pine species, they have not very big seeds. They have a wing. Actually, here the squirrels.
Okay.
Squirrels, they get a lot of seeds and make squirrel catches, they say, mountains. But I will think in pollen.
Pollen, okay.
Right? Pine species, you can see how.
Right.
Pollen can move even 300 or 400 kilometers if the thermal movements help. But 10, 15 kilometers, maybe for pines, is the common thing.
Okay.
Robledo Arnuncio in Madrid, in INEA, he has been studying a lot about this pollen theme. I'm not an expert, but pines, the genes can spread easily. Okay.
And the last quick thing was when you talked about costs of plasticity, which kind of costs were you thinking about?
Maladaptation. Plastic maladaptation. Some plastic response that actually doesn't increase fitness. And many of the plastic response are costly because, the organisms, for example, in trees, they produce new modules. So all the investment of this year has been, let's put an example in trees. They have the trunk, is the xylem, and all the water moves through the xylem in tracheids, that are in conifers, that the diameter of these vessels is very important for drought resistance because the tension of the water there is crazy. Under drought, the tension can be so crazy that makes empty spaces and the tree can die. So how the tree build this system is very relevant for resilience, and it depends on the environment in that point. But it's costly, right? Because some of the mechanisms of these vessels, if the tree is using osmolytes for regulate the things, or how the cell walls are built, can be costly, right? The tree can invest in making a very safe hydraulic system, but very safe hydraulic systems are hydraulic systems not very efficient. Here we have a trade-off. And probably very safe hydraulic system is more costly from the point of view of carbohydrates. So if there's not a drought, to invest on that is actually a cost. For example, or in defenses, it's more clear. If the tree produces defenses that are very costly from a carbon point of view, terpenoids, phenolics, they are defenses. Actually, trees, they work with constitutive defenses. They are always there, and inducible defenses, because it's so costly that they cannot have enough defenses. But the inducible defenses can be slow to produce, and for example, this beetle is faster. So invest in defenses is good if you are going to be attacked, and this inducible part is the part that is very related with plasticity, right? How they induce. And actually, we know there's genetic variation in the inducibility, and is negatively correlated with the constitutive defenses. Trees with a lot of constitutive defenses, they induce less, and just the opposite. Examples of these costs in forest trees. Thank you so much.
All right, cool. Well, I think we'll leave it there. It's just past 2 o'clock. So thanks again for your talk and for the really stimulating discussion. Next week, we're off. No more seminars for this year, but join us again next year on the 13th of January when we have Ana Travesat visiting to kick off a new season of seminars. Thanks for coming.